So far, we've talked about RNA that codes for stuff, but our RNA is going to be an example of a type of RNA that has a structural component, kind of like proteins. Our ribosomes are actually going to be composed of rRNA, or ribosomal RNA, as well as proteins. So it's a hybrid between rRNA and proteins. So our ribosomes are going to be composed of two subunits. It's going to have a large subunit and a small subunit. So here's the large subunit, here's the small subunit, and contained within that large subunit, you're going to have this enzyme, peptidyl transferase. So A's meaning it's an enzyme, transfer meaning it's transferring something, peptidyl it's probably going to be transferring, something to a protein involving a protein of some sort. So, and that's going to be part of the large subunit. So peptidyl transferase is responsible for forming those peptide bonds in between your amino acids to make the proteins. Another type of RNA is transfer RNA, which has kind of a structural component as well as a coding component. So transfer RNA, or tRNA, is what's going to bring those amino acids to your ribosome. So it's not only free amino acids that are floating around in the cytoplasm, you also have amino acids bound to their appropriate tRNA, and that's what's going to be picked up by the ribosome. And the tRNA is going to drop off those amino acids to be spliced together. So our tRNA is going to be composed of, it's pretty much its body consisting of an acceptor stem, which is what's going to bind to that amino acid. And then it's also going to have this coding region, or we would say the anticodon. So it's going to have a 3-nucleotide anticodon located on it, and it's going to have an acceptor stem located on it. Now this is a simple two-dimensional structure. Here's a three-dimensional structure, and then here's the space fill structure. So it's just kind of like a glob of RNA with a little bit of a coding region over here and then a little bit of a protein grabbing or amino acid grabbing region at the top. So we're going to have many different types of tRNA because you're going to have some tRNA dedicated to specific amino acids. So for example, your serine, which is an example of amino acid, is going to be carried by a specific type of tRNA. Same with your aspartic acid, which is going to be carried by a specific type of tRNA, and your arginine is going to be carried by a specific type of tRNA. So to look at the structure a little bit closer, your tRNA, it's going to have a 5' end, it's going to have a 3' end. This is where it's going to bind to the appropriate amino acid, and importantly, it's going to have that anticodon region. So this anticodon region is going to be the anticodon for your mRNA. So let's say on your mRNA you have CCG. Well, your anticodon region is going to have an affinity for this CCG. It's the anticodon. And so that's how, when your ribosome is chugging along from codon to codon to codon to codon, well, it pretty much waits for the appropriate tRNA carrying the appropriate amino acid to bind with its anticodon onto it, and that's going to allow it to shift forward to the next codon. The next codon or triplet of nucleotides that would code for an amino acid. So here is just showing, if you have a ACG, and we'll pretend like the ribosome is, right behind this, your appropriate tRNA carrying the appropriate amino acid is going to come, and the anticodon is going to bind to the codon, and it's going to allow that ribosome to shift forward. So it's going to drop off that threonine, and then this tRNA is going to, dislodge, disband, and it's probably going to pick up another amino acid. It's going to be recycled. And then that threonine stays behind and is spliced together with the following amino acid, every time the ribosome moves from codon to codon to codon. So here is a more definitive table, one that I'll probably use on exams or in examples. AUG is going to always be your start codon, so all of your proteins are going to start off with methionine. And so here, the way I would use this, or the way you should use this, is the first letter in your sequence. We could actually just use this as the example. So we have AUG. So the first letter, so it's labeled here, out of all of these letters, G, A, C, U, the first letter of AUG is A. And then you go up here, the second letter, what's the second letter? U. So we have U here. So the cross section between A and U is this box. Now within this box, we just find G. Here's G, methionine. So that's going to start off your ribosome. So next we have GAC. So we have G here with our first, which is our first letter, and then our second letter is A. So the cross section between G and A is this box. The third letter is C, and that's going to code for aspartic acid. Now you might notice here you have a little bit of redundancy. So aspartic acid is going to be carried by, your tRNA with the anticodon for GAU. It's also going to be carried by a tRNA with the anticodon for GAC, for example. AUA is the next one. So we have within our first letter A here, and then U is the second letter, and then the third letter is A, so AUA, and so that's going to code for isoleucine. Then next we have UGA, so we have our first letter here U, our second letter G, so the cross section between U and G is this box, and UGA, stop codon. So there we've translated from our mRNA to our protein, or our amino acid sequence. So you had methionine, and then we had aspartic acid, and then we had isoleucine, and that's it. You have a tripeptide. Tri meaning 3, peptide meaning having to do with proteins, tripeptide. All right, so ribosomes. Let's just talk about the ribosome. Remember, the large subunit is going to contain that peptidyl transferase, that it's going to have that enzymatic activity, and it's going to have 3 binding sites. So the T, or the A site, that's actually where the tRNA lands. We could probably just use the T site for the tRNA. So that's going to be where the tRNA will land on your, or make its initial landing on your RNA. So here you have UUC, or actually, we just move with that. Okay, so we got UUC, which is a codon, and your anticodon is going to be AAG. So tRNA, the appropriate tRNA with the appropriate anticodon for this codon here, carrying the appropriate amino acid, is going to come and drop off its amino acid. Then what happens is your ribosome shifts over, and it moves this tRNA carrying phenylalanine into the P site. So this is where the peptide bond is going to be forming. So peptidyl transferase is going to splice this phenylalanine to this tryptophan. And the thing is, when this AAG is moved into this P site, this ACC is going to be moved into the E site, and that's going to be kicked off. So this AAC tRNA is what kicked off, you could say, moved this UAC over from its P site to the E site. All right, so here's the small subunit, here's the large subunit. This is where all the action happens. So when it comes to translation, we have, kind of like transcription, 3 major steps. The first step being initiation. So what's going to happen is your small subunit is actually going to be bound, so the small ribosomal subunit is actually going to be already bound to tRNA carrying methionine. So remember, methionine, that's going to start off all of our proteins, right? So it's actually bound already to the start codon, or the start, you could say the start tRNA. So the small ribosomal subunit is bound to a tRNA carrying methionine. And what it does is it picks up that RNA, that messenger RNA, that mature messenger RNA, and it scans until it finds AUG, which is going to be the start codon. So once it finds that start codon, well, it binds to it. And remember, we only have the small subunit. So the small subunit waits around until the large subunit comes and bumps into it, and it forms the full ribosome at this point. So this large ribosomal subunit finds the small subunit, which is bound to the tRNA, bound to methionine, which has found the AUG, or the start codon. And then we consider this whole thing right here, at this point, the initiation complex. Initiation. All right, so next we got elongation, kind of like transcription. And with elongation, also like transcription, what's happening is we're just elongating that peptide sequence, or you could say that protein. So here I actually kind of like this figure a little bit more than the other one. So you have your tRNA binding to, sorry, actually you could say this tRNA binding to the T site, and when it does that, the ribosome shifts over, moving it into the P site and then kicking off the tRNA. So when it binds to the, you could say, oh, actually they call this the A site, whatever. A, T, it doesn't really matter. Probably not going to ask questions about the letter designations for these. So your tRNA bringing the amino acid is dropped off, and then peptidyltransferase is going to fuse these amino acids together by forming a peptide bond between them. So peptidyltransferase integrated into the large subunit catalyzes the formation of the peptide bond between the amino acids. So we've got the 2 amino acid chain here, which is then moved into the P site, which kicks out the old amino acid carrying tRNA, and then it moves on to pick up another amino acid and so on. It's recycled. Okay, so eventually, after all that elongation, what we get is termination. So termination is where we cease to translate the mRNA into protein. So eventually what happens is this ribosome is going to reach what we would call a release factor. So the release factor binds to the stop codon, binds to the stop codon and causes peptidyltransferase to attach a water molecule to the terminal end of the polypeptide. And that's going to cause it to release from the ribosome. So here we have the release factor with its anticodon for the stop codon. So the stop codon could have 3-letter designations, UAG, UAA, or UGA. And so you have that release factor carrying that anticodon bound to water, bound to GTP, which is another form of energy we can use in cells. So it actually costs energy to do this. And that's going to cause that ribosome to disband, and then freeing that polypeptide chain. So here's that polypeptide chain. So that's translation. So translation starts off with initiation, where the small subunit assembles on the mRNA and waits around for the large subunit. Eventually the large subunit binds to it, and then you have your initiation complex. And remember, the start codon is always going to be methionine. And then next we have elongation. That's just where your appropriate tRNAs, bound to their appropriate amino acids, come along and deliver their amino acids so that the peptidyltransferase within your ribosome can splice that together with the amino acid prior to it. And then eventually your ribosome reaches the stop codon, in which the release factor will bind to it and then cause that ribosome to disband. So in prokaryotes, it's actually going to be a pretty quantity over quality kind of organism, where transcription, so as transcription is happening on the DNA, your ribosome, so like as you're creating that mRNA, instead of splicing it and then sending it out of a nucleus, remember your prokaryotes don't have nuclei, your ribosomes are already going to be binding to that mRNA, and it's going to be translating that mRNA immediately. So transcription is the same process in prokaryotes, but as the mRNA is being created through transcription, translation begins already. So here it looks like this is the first period, and then as it's creating that longer and longer mRNA, it's actually creating all of these little brown globs are actually ribosomes that are translating it already. Okay, so this is important to know. Your RNA is made in the 5' to 3' direction. So it's made in the 5' to 3' direction by RNA polymerase. And if it's making it in the 5' to 3' direction, if you remember, our DNA is anti-parallel. So if it's making it in one direction, that means it's going to be reading it in a different direction. So it's going to read DNA from 3' to 5', but it's going to be making RNA in 5' to 3'. And then mRNA is going to be read by the ribosome in 5' to 3' direction.